Hi. My name is Lakhani. I run a consultancy in London, and currently I'm working at an investment bank. But for various reasons, I thought I'd give you a talk about kind of the use of Spark and big data in life sciences. So in this talk, I'm going to give you a bit of a background into Apache Spark, which is kind of taking off. You've heard a lot in this conference about how Spark is trying to replace Hadoop, and I'll give you some of its applications to life sciences. So on the example on the right there, you have an electron's image of a cell. Now, it looks pretty innocent, just looks like a bunch of matter. It looks like a few kind of interesting structures within it. But from a data perspective, it's actually a huge problem. As I would say, in terms of the volumes you get of just trying to represent that cell in a data format, it's huge. It's the size of Google, probably double, probably triple, which is why Google is getting interested in these kind of problems. So something like Spark makes it really easy and a kind of a natural fit to use in life sciences and in kind of genetics and systems biology. So what I hope to do in this talk is just give you a couple of use cases of how you can use Spark for life sciences. But the application extends beyond bioinformatics and science. You can see how you can use Spark for other applications as well. So just a quick introduction about me. I work in banking, I worked in retail, I worked in a number of industries, but my background has been in software engineering and computational biology at Oxford. So it's from this beginnings that I started to do consulting in other industries. So here's a typical snapshot of the data we have today. It's a snapshot from a publicly available database. So you can see that we've got 60,000 genes, 19,000 proteins, and so on. On their own, these figures don't look like much, but then you think about the number of combinations that are taking place. So 19,000 proteins can interact with any of those 60,000 genes, and then you've got the whole table below it. So the complexity of this whole problem just becomes really huge. And then to make things worse, you've got population, you've got differences in populations which confound this issue as well. So people in Africa will have a different set of, a genomic structure to people in Asia. And then if you're trying to create a drug for the market, for example, you have to take into account all of this complexity, and it becomes a huge big data challenge. So what's the speciality of using Spark? Well, Spark is a distributed processing platform. If you've not used Spark, it's favored towards in-memory processing. Okay, so you have things like Hadoop, which is take all this data from clicks and banks and all this sort of standard data that you get. But Spark is more designed to be in memory. It's designed to be more streaming oriented, and it's got a good framework for machine learning that's built on top of it. You may have heard in the last couple of days, you've got various parametric, non-parametric models for doing kind of machine learning. So for the life sciences, it works quite well. And then what's kind of growing is this field of deep learning that's a layer above Spark. Okay, so there are a few kind of providers who are building deep learning packages on top of Spark, which help you do this kind of analytics at scale. So one of the kind of the new features of Spark on top of Hadoop is this concept of broadcasting variables. In Hadoop, nothing is shared between all of your workers. Okay, so if you're doing a kind of a job, it's the map-reduce paradigm, where you send some data out, you do some aggregation, and you bring it back. But Spark has this capability to have a broadcast variable, which means you can share data amongst worker nodes, and this is quite useful for kind of genomics. And there are various graph libraries and text processing which kind of help with the sequence analysis and the genome analysis that we need. The main strength of Spark is that you can use something called a resilient distributed data set. So as long as you kind of conform to an interface, you can bring in any data from wherever you want. Okay, so you can bring it in from various sources. And traditionally, the kind of the genomics, the academic community is not very good at standardizing the data sets, but there's sort of a consortium to bring this all together, but using RDDs, you can overcome that problem. So this is a typical use case in kind of modern day bioinformatics. You want to find out in a particular experiment how much protein is being made by given an experimental condition. So one of the ways you do that is you measure the amount of RNA in the cell. So on this bit here, you chop up the RNA into what's known as reads, and the reads are fixed length. So you kind of have an experiment which breaks this up into standard reads, and then you have a reference genome, so a genome that's been kind of standardized by the community, and you try and map the number of reads to the genome. So that gives you an identification of how much protein is in the cell at a given time. So in itself, the problem seems straightforward. Okay, you got reads, you map them to a genome, what's the big deal? The problem is you have thousands and millions of reads under different experimental conditions. The data volumes for one experiment, for example, just one kind of read is 5 gigs. So if you imagine you've got experiments from thousands of universities under thousands of conditions, this data grows very quickly. The other kind of aspect is you want to do this process very quickly. So sometimes you want to do almost real-time monitoring of experimental conditions. So you want to be able to do these read alignments very, very fast. And so this is where Spark and distributed data comes in very handy. So what you get at the end of this is you get these aligned reads, and they give you an indication of how much protein is in the cell. So here's an example of how you can use Spark to do that. So you have a broadcast variable, which is your reference genome. This is the thing you're trying to align to. And on each worker node, what you're going to do is you're going to take those reads and then you're going to align them to the broadcast variable that you have here. So there's your broadcast variable. Those are your reads, and you're doing this all in parallel. So even if you have billions of reads, you can do this very quickly and get quantification quite fast. The next aspect is, well, traditionally Java's not been a good language for bioinformatics, primarily because of these overheads of the garbage collection, and things like the string object has a lot more overhead than in other languages. So you've got DNA, you've got base pairs, ATGC. If you're representing this as strings in Java, there's a lot of overhead, and that kind of slows things down, which is why people prefer things like Python and other languages. So what people at Spark decided to do was almost go back to the drawing board and look at these use of unsafe objects, which are more C-like and direct memory access. So rather than using the Java representation of strings, you can write off-heap strings, which means they're not prone to garbage collection. You're not waiting for the GC pauses that you get with traditional Java programming. There's a lot of other features the Spark developers have put using this unsafe kind of packages, which means that you've got more faster native processing for big data applications. So here's an example of what's known as, you could say this is a start of systems biology. So on this line you have some DNA, which then makes some proteins. Proteins interact with each other, and you have sort of other microRNAs that bind to the protein and then inhibit its function in some way. Okay, so it upregulates or downregulates a particular protein, and you get this huge complexity of things that happen. So let's say you've had some lunch, you're getting some insulin response. All these things are coming into play, and it's for scientists to figure out what's going on at what period of time. It becomes a huge challenge, you can see from a computational point of view. It's a big data problem, not in the way you've seen before. You're not trying to quantify clicks. You're not trying to do the standard retention of customers. It's a very different type of problem that you're facing here. So the one way you can do this is by representing the data as a table, as you do with everything else. So on here you have one interactor, so one protein. You know the counts. So you've counted the amount of mRNA that you've produced. You put it in compare it to another table, and you can build up this profile of different things. At the end, you want to then figure out for a new protein what that expression level is going to be based on 2 other proteins, for example. So it's a bit like customer recommendations. 3 people bought this. This person is new. What does he buy? Okay, it's the same kind of thing, and our good old friend deep learning comes into play here. So if you're not familiar with deep learning, you can summarize it as you have a set of inputs on this column here. So inputs are the actual expression level of a protein.
you have this magical middle layer, which is what the algorithm tries to figure out. It tries to figure out all these different combinations of weights, how much weighting to give to a particular input. You apply the softmax function to this whole thing, and you get some outputs, which is what the prediction is. Okay, so you're trying to predict expression levels from this set of inputs. This is used everywhere. I mean, you use it for, all the kind of use cases you've heard over the last few days. There's a lot of interest and exciting kind of potential in this, but in my opinion, it only works when you have lots of this data to train on. And in biology, if each experiment is costing you, $5,000 just for one set of inputs, you're going to need the community to bring this aggregate picture of experiments together for you. And this is where you use Spark and use open data and you use these kind of frameworks to bring these models into play. Otherwise, you're going to be sitting in your lab with your one experiment. It's not going to help anybody very much. So there are a few frameworks that we use for deep learning. There is deep learning for Java. Again, they're coming up with models to build on top of Spark. Okay, so you can use this on a standalone machine. You can try and practice your models out on your laptop, and then when you're ready to ingest data, it easily scales out without much change. Google's just open sourced its TensorFlow. I haven't had much experience with this, but it's C++ and Python based. But there are, the kind of libraries for Java that are coming out soon. Another really cool framework is H2O. If you're kind of a non-technical person and you just want to get started with deep learning, they have a really nice front end. So there's like a web UI. You kind of click and point and give it your data. It'll build your models. You set some parameters, and it'll give you some nice predictions. So in each of your use cases, if you just want to try out some machine learning, then I'd recommend you try H2O. Okay, so another kind of use case is protein folding. So each of the proteins in our cells, they start out as these individual sort of lines, and they kind of, in nature, they just naturally fold to this conformation state, okay? They do it using the laws of physics, but to simulate those kind of mechanisms is really complex, and it's a huge challenge. So there's another example of how you can use Spark. You can kind of use Spark's distributed processing to go and try out loads of conformations, try out different combinations of how this protein would fold, and then come back and give you the best answer. Finally, you've got some use cases with ontology mining. Scientists are very good at kind of, for each protein, they give it a label, so they say this is what the protein does. So you can use Spark to build up graphs of these kind of ontologies, which you can use the graph libraries in Spark to find, say, represented terms. So this is good for textual analysis, okay? So if you want to just figure out which proteins are involved in which functions, you see reds here, they become good candidates for drugs, for example. That's just another example of how you use Spark, the graph kind of libraries, where you can build up these profiles of the cell, so you try and make meaning of the cell just by exploring the various relationships between the ontologies in the graph. So there are lots of other use cases. This is just one example. Virtual drug screening, where you can screen thousands of molecules against a protein to find drug candidates. This is what pharmaceuticals are really interested in, because it costs them millions of pounds to try and find drug candidates. Genome-wide association studies, as I mentioned, you've got different populations, so you want to try and find differences between the populations and so on. And finally, so we had a quick introduction to Spark. We can use the scale-out computing model and broadcast variables for kind of sequence analysis. There's some good machine and deep learning methods available on Spark, which will help the scientific community. And you can leverage the graph libraries for ontology mining. Thank you.